Good morning, everybody. Thank you for attending. My name is Anna Marie, and I'm the Marketing Director here at NECi. Been here for about 2.5 years, and I've played multiple different roles here, from working in the lab to marketing, and I also help out on a organic farm locally. So I do work with the soil sometimes in the summer. And here with me I have Ellen Campbell, and she's the CEO of NECi.
Hi, we started NECi for about 23 years ago. My husband and I were doing academic research on nitrogen metabolism in plants and found a way to use nitrate reductase from corn to measure nitrate in anything. And that's one of our basic foundations of our company and why we know a lot about plants.
So here's a quick agenda. We'll just give you a few seconds to look over this, but basically we'll be going over two main topics today. First, we'll be going over the philosophy and reasoning behind nutrient analysis in your soil and how this benefits soil health. And then we'll be going over the procedures and methods that we've come up with to help you analyze nutrients in your soil.
And this webinar will be available on our website, so you can look at this again anytime.
And if at any point you have any questions, please feel free to put them in the question box and we will answer them at the end of this webinar. So we'll be focusing on two of the most important macronutrients when considering soil health, which are nitrate and phosphate.
Okay, what we're going to be talking about are the forms that are available to the roots right away. So we're talking about the water extractable, free nitrate in the soil, and free phosphate or orthophosphate or reactive phosphate. There are many different terms for that. We're not talking about phosphate that's bound to soil particles. We're just talking about phosphate that's free in the soil. Nitrate is extremely water soluble. It doesn't bind to anything. It moves when the water moves, and that's one of the reasons nitrate testing throughout the season is critical. Phosphate does bind to soil particles, and so its availability changes with soil chemistry. Both of these, you're never going to get good yield if you don't have adequate nutrition.
So historically, the strategy for maximizing crop yield has been to apply an abundance of nutrients to farm fields without necessarily considering the consequences of over-application. And what's been discovered is that that is not a sustainable practice, and the more nutrient application doesn't necessarily translate to a better outcome. And we've seen the results of this in Lake Erie and in the Gulf and in other coastal regions where agriculture is important. And so over-application of crop nutrients is a source of concern from many viewpoints. If you are applying these nutrients to a field and they're running off into the water, then they're not in the field, and this means that there's fertilizer being wasted, which means there are economic resources being wasted. And with the low profit margins forecasted this year, it's even more important. And also with a growing population, there's an increased need for food security and therefore a need to maximize yield, ensuring that our land is being used the most effectively. And again, from a water quality standpoint, nutrient runoff raises human and environmental health concerns. Again, this is what causes algal blooms in Lake Erie and the Gulf and in other spots. And also private wells are located in agricultural areas and they are unregulated by the EPA. Therefore, nutrient contamination can often go unnoticed in these places. The US EPA Clean Water Act sets the maximum of nitrate in drinking water at 10 parts per million, and elevated levels of nitrate in drinking water can decrease oxygen carrying capacity in the blood, which is what causes blue baby syndrome. And removal of these nutrients once they're in the watershed is very difficult and also very expensive once they're in the groundwater, as we've seen in the case of the Iowa Waterworks lawsuit. And from this lawsuit, There's, in other situations similar to this, there's often a stigma that these two sides are opponents, but really the truth is everyone's concerned about these issues, and it's not a battle because everyone benefits by limiting nutrient runoff, from farmers to environmentalists to recreationalists and homeowners. Food security, sustainable soil, clean drinking water, and healthy ecosystems can all be achieved, and everyone's going to be happy by managing nutrients at their source, which is the soil. And so these are some of the different strategies that farmers are implementing to help with nutrient runoff, and there's many different organizations trying to push these strategies. And there's more information about these at the end of this webinar if you'd like to learn more about these. And so how do we know that these strategies are working? Through nutrient analysis. By managing nutrients, knowing how much is in the soil, the proper amount can be put down so that there's no waste. And so when you're testing your soil, there's going to be many different variables, from the depth to the time of year, also rain events and your land capability class. So depending on what you're growing and where you're growing, it's going to dictate how you're going to want to test your soil. And so here is the land capability class map. As you can see, the very dark green has the most fertile soil, and that's classified as number 1 land, and it progressively gets less and less suitable for crop cultivation here. So again, we'll have these resources available at the end of the webinar. So the 8 classes are subdivided into capability classes, again 1 through 8, 1 being the most suitable for crop use, going down from there. Between 1 and 8, there are subclasses which go into climate, erosion, wetness, and shallow soils, and some of the classes that the NRCS has identified as important for nutrient management include the ones that are listed towards the bottom of this slide. So a couple different ways that you can see if your soil isn't adequate in nutrients, you can see from the plant leaves. So if the leaves are dark or purple reddish, that usually means there's some sort of phosphate deficiency. And with nitrate deficiency, the leaves often will look somewhat transparent, very yellow and pale. This affects their ability to produce chlorophyll, so the results are very visual here. And again, these nutrient levels can change over time, so one day the content could be a certain number, and with a rain event it's going to differ the next day. This also is affected with how the plants uptake those nutrients and the water table fluctuation. So a good way to prevent these things from happening is through soil nutrient analysis.
Where am I supposed to? Okay. What we started, the USDA has been funding our research and developing these tools because we started looking into this issue about who needed to measure nitrate, and we saw that the tools available for managing nutrients were really not adequate, especially for users that were on site and in the lab. So we've developed new tools for nutrient analysis based on enzymes. We feel that if you're going to tell people to test and to have good control of what they apply, they need to have numbers that mean something. The numbers need to be valid. So we've developed the lab test first, and then we go on to develop on-site tests that anyone can use so that your results will match, so that your on-site results will match your lab results, and you can make better decisions and more timely decisions on when to apply and when not to apply. Are your buffer strips working? is it time to add a new PSNT testing for corn is very important. All of these timely decisions need better data. What we start doing is we find an assay that needs to be done, and then we find an enzyme that does it, and then we make a recombinant version of that enzyme. We validate these methods very strongly. We'll show that later on. And then we develop the lab methods first, and once we know that the method works, then we simplify that down for the on-site methods that anyone can use. We want these methods to be accurate, reliable, and selective, meaning that you don't need to do a lot of sample prep. The enzyme will find your analyte in a very complex mixture. The other advantage of enzymatic methods are they're environmentally benign. There are no toxic reagents in here, especially for the field kits.
So now we'll go over how these procedures actually work, from collection to preparation and then testing them. So soil sampling is going to depend mainly on depth, time, and then quantity and pattern. And this isn't something that we're really going to get into because it depends on your region. So stick to what you know for soil sampling. If you need advice, you can contact your local agricultural extension or crop consultant. But we will say that it is best to test the soil immediately after collection. If this is not possible, try and keep it cold. And if you're in a laboratory, or if you would like more precise analysis, you can dry your samples to constant weight, and we have protocols available for this, and we'd be happy to send those to you. They're also available on our website.
For extraction, we're only extracting with water here because, again, we're looking at the nutrients that are immediately available to the plant at that moment, and they're only going to be available in water. You take a gram of your soil, shake it in 5 mils of lab quality water, which we supply in the ag kits. You let it settle. If you need to filter it, you do. You're only going to be taking 150 microliter drop of this extract in 1 milliliter of buffer. So it's a 1 to 20 dilution. So color, pH, a lot of potential contaminants, everything's diluted away. This is another advantage of our enzyme-based methods over more chemical methods. You let the reaction run for 10 minutes. The reaction goes to completion, can't go backwards. Once you get nitrate reduced to nitrite, it's going to stay as nitrite. You add the color developments, and you can either see the results visually. We provide a nitrate standard with the kit in the center of the range, so you can compare is it higher or lower than your target. We also have a free nitrate color slider available on our website for Androids or iOS. And then we now have a new photometer, so if you want digital results with recordable data, and we'll go into that later.
So the first step in this is extracting the nutrients from the soil. So again, you'll take a scoop of soil, which is equal to about a gram, extract it in about 5 milliliters of water, mix it, and then just set the tube upright to let it settle. However, sometimes this won't be possible with certain samples that are high in clay or silt content. So centrifuging, using a syringe filter, and also just a basic coffee filter are all great methods for filtering these samples, and we have these protocols available on our website, and we'd be happy to send those to you after this webinar for more detailed information regarding filtering. And so now we're going to go over nitrate analysis using enzymes. So after you extract your sample and filter it, you're going to add a drop of the sample to the cuvette, which is shown on the left-hand image on the bottom. This cuvette contains the enzyme and the cofactor needed to make the enzyme work. In this case, it's nitrate reductase, and it will convert all of the nitrate to nitrite. After about a 10-minute reaction, all the nitrite in the sample is going to react with color reagents, which is shown in the third image. It's just a powder that you add, and this is going to turn the sample pink. So the darker the shade of pink, the more nitrate in your sample. So this can be analyzed visually with the color slider, or it absorbs at 540 nanometers. So if you have a spectrophotometer or one of our handheld photometers, then you can get digital results from this.
We're just showing you the reaction for people who are interested in the chemistry. For the lab people involved, basically nitrate reductase replaces cadmium in the standard methods. It's after that everything's the same.
And again, this is available on our website, and we'd be happy to provide more information about this. And so now we'll go over the phosphate method using enzymes. This image is the graphical abstract from our recent publication in NECi this past summer. It's similar to the nitrate method, except there's no color development, and it's not visual analysis, so you will need a photometer to analyze the results. But as you can see, it's a pretty simple method. You just mix your soil with water to extract it, let it settle, and then add it to the enzyme mix, and after a 10-minute reaction, you can see how much phosphate is in your sample.
This is just some data from the NECi publication. NECi is a peer-reviewed journal, online journal from Elsevier. We purchased some standard soils from ALPS and just showed that, yes, our numbers obtained from our assay match within a very good range. The standards, and then we also did spiking, where we add a known amount of nitrate to the sample, and then we see that we recover 100% of what we added plus what was there in the first place. These are standard validation methods that are used for anything for chemical validation. Again, here's the chemistry of the phosphate method. This method's been used in medical analysis for quite a while, and we just adapted it for agricultural type samples.
So if you're in the field, you will need to use a photometer to get the phosphate results, and for nitrate you'll obtain digital results instead of analyzing them visually. So we've just came out with our new handheld photometer, which interfaces with mobile devices. All of the software and the brains are on the mobile device using an app, which is what does the calculations and tracks and stores the data. So you can see on the right-hand side, it's just a screenshot of the app being used to analyze soil for nitrate, and it's just easy as clicking the button. On the left, you can see the photometer. This accepts a standard 1 ml cuvette, and so that's with the flat sides, and this increases accuracy when compared to other commercially available photometers, which use either large volumes or round vials. In the app, there's also options for time and GPS tracking, and these interface with each other via Bluetooth, so you don't actually have to connect them through any cords, so you can take this device anywhere, and it has a pretty long battery life, so you can analyze your results on site in the field.
And the app stores everything in tab delimited format so that it can go right into Excel and interfaces with any of the standard ag packages, FarmWorks, AgWorks, any of these ag packages. Our data will shoot right in and happily function with all of your other applications. Our philosophy of developing methods is we design and validate the lab method. We didn't show you any publications on the nitrate method because that's been around for 20 years. And we have standard method status with ASTM International, with the U.S. Geological Survey, and we're about to get standard method status with the EPA. We then validate it for all the different kinds of samples that are used for agriculture, because there are no standards companies that validate for ag samples. So we get corn plants from all over the place and soil samples from every place and make sure that it actually works. For those on-site kits, then we have everything pre-measured for you. So you just open everything and pour. You don't have to think. We pre-calculate, so if there are dilution factors, they're incorporated in with the digital readout that you see. So, or in the lab kit and the kit instructions. So you don't have to try and remember, we don't even use the term dilution. You'll never see it. We've done all that for you. We've carefully selected all the components, so we bought every kind of 1 gram scoop available, and we saw which ones were the most accurate. We also checked to see that that scoop that we have, 1 gram of any kind of soil from sand to mud, really pretty much weighs a gram. The variation isn't worth worrying about for a field kit. And same with drop pipettes. We bought everybody's and found the one that was 1% accuracy. And again, everything's the best quality lab consumables that you can buy, and because we want you to have good results. We also want to let you know that these kits are stable enough that you can leave them in the dashboard of your truck for, say, a couple of days, and they'll still work. We guarantee a week at hot summer weather.
So again, we provide laboratory and on-site kits. Like Ellen was saying, the on-site kits have been made so that there are no calculations or measuring involved. Everything's pre-measured and pre-calculated for you. The laboratory kits are the same reagents, just formatted for lab users. And we also have reagent packs for more economical analysis for experienced users and users who are doing routine automated analysis.
So we have microplate format, we have reagents for different discrete analyzers. We've worked with most of the manufacturers of auto analyzers to package the reagents for their equipment. So just let us know if you have a way. We can adapt these reagents to any kind of format.
So here are some certain resources that we found helpful, and a lot of these resources we use to collect certain information for this webinar. So these will be available on our website if you want to check any of these out. And I want to thank you for attending this webinar with us today. If there are any questions, we can take those now.